So I hope the student, please you maybe apa, diperhatikan, and please maybe I want to our student untuk mencerna ya, atau menelaah, atau maybe if our student have question, maybe you want discuss of these activities. Uh, Kenapa ini menjadi sangat penting? Sekali lagi, untuk mahasiswa saya ini mungkin semuanya pakai bahasa Indonesia, tolong diperhatikan, ini menjadi suatu hal yang sangat penting. Why we reduce of a climate change, untuk mengurangi impact dari climate change itu terhadap aktivitas fisheries and marine science, itu menjadi suatu hal yang sangat penting. Itu yang pertama. The second, I hope this public lecturer give maybe sharing knowledge gitu ya. Kemudian sharing information uh, untuk kita semua gitu ya. For us, agar kita mendapatkan tambahan keilmuan gitu ya. I have a give added value for knowledge, maybe dari ini kan dari Jerman ya, apa dari mana Mas Budi Rudi? From Jerman ya?
Yes, saya from German.
Ya, ya, from Jerman. Jadi mungkin nanti akan dapatkan tambahan keilmuan tentang bagaimana pengelolaan biodiversity untuk mengurangi atau eh, mengurangi impact dari climate change. Oke, itu yang pertama. Yang kedua, and the last, sekali lagi saya ingin mengucapkan banyak terima kasih. For the ones again, I would like to thanks the speaker, the coordinator of Aquatic Resource Management Program, the chairman of Public Lecturer Committee, gitu ya, yang telah berjuang untuk melaksanakan kegiatan ini agar bisa sukses. And the last, with saying Bismillah, I declare this public lecture is open. Terima kasih, thank you very much seluruhnya kepada Bu Diaruri, kemudian Bu Maria yang sudah berjuang untuk membantu kita semua, kemudian juga pada chairman dan semua panitia yang ada di sini. Terima kasih. Bila begitu ya, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much to Dr. Endang Helmi, Master of Science, to give us a great opening speech. Move on the next agenda, we will have a photo session. Please, all of the participants, turn on your camera and give your best smile and icon, and I will account. Okay, the committee, please help me to screenshot. Okay. All of the participants can turn on your camera. Participant can turn your camera. Okay, the committee, please help me to screen the camera. I will count 1, 2, 3. Once again, 1, 2, 3. Once again, 1, 2, 1, 2, 3. Okay. One. Okay. Enough. Thank you for all the participants. Thank you for all the participants. Uh, uh, Before we move the main session, we would like to inform the participant. During the public lecture, there will be many voucher that will be given to the most active participant. So be ready and give your best question to the speaker. Okay. Ladies and gentlemen, let's move to the main session, the public lecture. This session will be led by our moderator, Mr. Ahmad Malik Firdaus, Master of Environmental Science. He is a lecturer in Department of Aquatic Resource Management, Fisheries and Marine Science, Jenderal Sudirman University. Without further ado, let's invite Mr. Malik Firdaus. So, Mr. Malik, the screen is yours.
Okay. Thank you very much, Ms. Dika, who has invited me as moderator for this topic. Ladies and gentlemen, may I have your attention? This presentation is about modeling the species diversity distribution and climate change, strategic implementation of MPA. Let's begin. The Honorable Mr. Chaya Chaudhary, distinguished guests, ladies and gentlemen, good afternoon. Sorry. Hi, doctor. How are you?
Hello, Mr. Malik. I'm very good. Thank you. How are you?
Fine, thank you. The Honorable Dean of Fisheries and Marine Science Faculty, Jenderal Sudirman, Dr. Ndang Hilmi, the Honorable Speakers for Alfred Weiniger Institute Bremen, Germany, Miss Dr. Chaya Chaudhary, the Honorable, the Coordinator Department of Aquatic Resources Management, Fisheries and Marine Science Faculty, Jenderal Sudirman University, Miss Cecilia Rani Samudra, and distinguished lecture staff, all participants and student of Fisheries and Marine Science Faculty, Jenderal Sudirman University. Before the presentation begin, I just want to welcome everyone to the webinar and thank you also much for joining us today. My name is Abdul Malik Firdaus. As mentioned, Ms. Dika, I'm lecturer in here in Aquatic Resources Management, Faculty Fisheries and Marine Science. I will be responsible for hosting this presentation today, and I'm glad to welcome Dr. Mrs. Chaya Chaudhary, who will present about MPAs and climate change. All right, without any further ado, I would like to welcome. And before we started, I will, if you would like more information about the presentation, it will be shared for you, and you can access in our website. And then please to screen the CV of Mrs. Chaya.
Yep.
Okay. Thank you. Wait. Okay, I just will to read the curriculum vitae with Dr. Mrs. Chaya Chaudhary. Her affiliation is postdoctoral researcher, Alfred Weiniger Institute Bremen. Email and nationality is Indian. Miss Chaya, you are Indian and you stay in Germany, right?
Yes, I live in Germany and I am from India. Yes.
Okay. Good. Professional experience, she's got postdoctoral, 3 postdoctoral. Wow, amazing. First in Alfred Weiniger Institute Bremen, 2021 until present. Second is postdoctoral researcher, Goethe University and Salzburg Natural History Museum, Frankfurt am Main, Germany, and postdoctoral research in University of Hong Kong in China. And he got PhD in Marine Science in University of Auckland in New Zealand, and Master of Science in Environmental Management. And I think she has many publication, and it's good for us to learn from her. And I think we can just start our presentation. Miss Dr. Chaya Chaudhary, time is yours.
Thank you so much, Mr. Malik, for the very nice introduction of mine. And I'm very humbled to present my work at this event. And it is really honoring. And thank you so much, everyone from the Faculty of Marine Science at the university. So I'm quickly gonna share my screen now.
Okay.
Yeah, can you see my screen?
Yes, we can see it.
Yeah, thank you.
Yeah.
So I will talk about my work on species diversity distribution and the modeling techniques that I use to identify the diversity patterns, and also what are the implications of these modeling techniques in real world, especially in the times of climate change. So I will take you through a journey. As Mr. Malik mentioned, that I have did my PhD in New Zealand and then moved to Hong Kong and then to Germany. So it has been quite a ride from India to Germany. And as the years have progressed, I have also gained a lot of experience in this, and it's I work in biogeography, and moving from places to places, this just teach me a lot about the environment, the culture, the biodiversity, and I'm gonna start from the beginning, how I got into this field. And so I'm going to talk about the very basics of the biodiversity analysis. That begins from the species diversity indices and what spatial scale that we look at, how we extract the data and how we process that. And then the spatial distribution of species diversity, how the species diversity is distributed in space and also in time. And this space includes latitude, longitude, and depth. But in this presentation, I have only included latitude and longitude. And we'll see why that is so important in the times of climate change, and how these diversity patterns have been shifting and will shift in the future. And how these biodiversity models are so important, especially when we talk about the implementation of marine protected areas. So we begin from the key concepts. So if you see in this diagram, there is this these symbols, which we consider as different species. So different symbols are basically assumed they are different species, and which are present in a smaller circle, site A and site B. So when we look at a very local scale, for example, how many species are present, what kind of species are present, we talk about alpha diversity. And then when we compare two sites, especially in terms of species composition, for example, if I go from site A to site B, how the composition of species is changing. For example, if there is completely like 100% different species found in site B, then we say that there is a very high turnover of 100% because all the species have changed, even if the number stays the same. The other option is when the sites are nested within each other, which means that, for example, you find some common species among these these two sites, and then we can say that A site is nested within B site or vice versa. And the diversity index that we generally put all these things into is called beta diversity when it comes to the comparison between two different sites. And now we zoom out a little bit and we look from a bigger perspective, where these little small local regions are in a bigger region. And when we maybe, let's say, count all the species within this bigger region, bigger circle, then we call it gamma diversity. So basically, then it will be like how many total number of species are there, for example, in this one big region, and that accounts for gamma diversity. And this is the base of biodiversity analysis, I would say. And we move into the next slide. And then my work has so far been at a global scale, which means this whole world is my laboratory. And I've been just looking at how the species diversity has been distributed if we go from north to south or from east to west. And for that, I divide these bigger regions into smaller regions and into smaller regions. So so you can see that at regional scale, when we see that this Earth is divided into hexagons and smaller hexagons to finally, if you want to zoom in and see the better resolution. And if we just talk about the latitudinal bands, just it depends upon the objective of the research, basically. So if if we are more focused into the local patterns or regional patterns, then we look at finer resolution, for example, these hexagons. And when we look into larger or general patterns, for example, latitudinal gradients, and considering how much data we have, we put the data into these grids and see if the results, what we get, and the analysis give us a statistically significant answers, and then we do the analysis based on that and get the results. So you will see. So I say these are 5-degree bands, the big rectangular in figure A that you see. And then inside of that, there are these hexagons for this scale. And this I started in my PhD. And these hexagons are really big. So I think the smallest one is 50,000 kilometer squares. And these latitudinal bands are actually 5-degree 5-degree latitudinal difference is 5-degree band. So for example, if we see from north to south, we divide the Earth into 180 degrees, and then by 5, so I would say by 90 bands, with a difference of 5 degrees. So these are really big ones, but it helps us in identifying the generalized biodiversity patterns. You will know what I'm talking about in just a few minutes. So this is just the overview of what we are looking at. So in alpha diversity, we are looking at hexagons, which are 50 kilometers square in size. And there we look at species richness, which is basically an estimate of number of species. Then gamma diversity is at a bigger scale in 5-degree latitudinal bands. And then beta diversity is basically just the species turnover, how the species composition changes when we go from one latitudinal band to another, or one hexagon to another. And then we use different indices for that. So we use different global databases. For example, Ocean Biodiversity Information System, Global Biodiversity Information Facility. And these are basically really large repositories for species occurrences, especially for marine. And GBIF also has terrestrial information. So I have mostly used OBIS for my research so far. And these are online available databases, and you can just go on this website and download the data for any species, for marine species, looking at where they occur. and what are the environmental parameters at which they occur. And if you are lucky, then you will find also a lot more information on their taxonomy and on their life stages, etc., etc. And then we basically cross-match, or because this data is just coming from all the resources, from the research vessels to individual observations to experiments. So it is very important to quality check the data that we are using. And we do this especially because there are a lot of synonyms and misspellings of species names and things like that, and their taxonomic classification. So to just make sure that what data we have is a quality data and is correct, we cross-match the species names with this World Register of Marine Species, and also extract more information about their habitat and their taxonomic classification. And then we use environmental data from Headleys, which is basically a sea surface temperature and oxygen. And there are also other databases like BioOracle. They also provide the RCP scenarios for the future, that were proposed by the IPCC. And GEPCO is just like provides the depth layer for the whole world. Yeah, so just for an example, OBIS at the moment hosts like 122 million records of species. That's a really big database. And we can see that there are like 183,000 accepted taxonomic names here. Yeah, so this was the beginning of my research, because so this was the beginning of my research, where I just arrived in New Zealand in 2014, and there was a controversy around because my senior Hania Saidi, she was finding a pattern which was a bit different from what was actually being said. So she was looking at the latitudinal gradient, or how the species diversity of razor clams changes when you go from south to north. Yeah, so the general hypothesis was that all the species basically, the diversity is the highest in the tropics. So when you go in the tropics, you find more number of species. And then when you go on the higher latitudes, the species number declines, which means that we should have a peak at the equator, right? Which is called a unimodal pattern. But what she found something very different. I mean, it could also be asymmetric unimodal. This is another hypothesis which many papers have reported, that the peak is not at the equator, but on the either side of the equator. But there is only one peak, they said. But then she was finding something very interesting, which was like a species richness dip at the equator. And the peaks were at the subtropics. And this could be asymmetric or symmetric bimodal. So we had like 4 possibilities of this latitudinal gradient in marine species diversity. And my work was to find out, first going into this OBIS database and finding out what the raw data says, and the other work was to figure out the data from literature that have also found similar pattern, but they have just not reported that, or they have said there is a sampling bias, or they found something unusual, which is not in line with the original hypothesis. So I ended up collecting the data and plotting the species richness against latitude. So in this table, you will see that on the first left-hand side, there is different taxonomic groups that I looked into from different papers and also OBIS. And this is the total, next to that column is the number of species that we looked into. And then we go from south to north. So the minus sign indicates southern latitudes, and the other ones are northern latitudes. And the gray color represents the dip at the equator in species richness, and the black color represents the peaks in species richness. And you can clearly see that there was a very common dip at the equator in all the taxonomic groups. So it was very clear that most marine taxa that have been studied, or for whose the data was collected, they all show a diversity dip at the equator. But there are peaks on the either sides of the equator. And this review was then published in Trends in Ecology and Evolution in 2016. And then there was a lot of controversy around that, like why it is like this, or it is just because we have less number of samples at the tropics. So then my work was to actually dig deeper into the data and find out if this pattern is significant and the dip is significant. So for this, I worked with 23.7 million records of species distribution from OBIS, and then did the quality check, and finally had a distribution for 51,670 animal species, and that had 12.6 million occurrence records, and these were collected after 1920. And then I defined these data as these animal species as pelagic and benthic based on their one. If they have one life stage as benthic, then they were considered as benthic. And if they had all life stages as pelagic, they were considered as pelagic. So now the task was to first correct this data for sampling efforts, for the number of samples. And for that, I used Chow index. And Chow index is an estimate of species richness that also accounts for the number of samples. So we know that our data to an extent is not biased by the number of samples. And we did this modeling, used this distribution against latitude model using generalized additive models, and found out that almost all different taxonomic groups showed a very significant dip at the equator. So these graphs are basically the same way that goes on the x-axis from south to north. And on the y-axis, there is this species richness. So you can see all the species here have most of them have symmetric bimodality, but also some of them has asymmetric bimodality. Nevertheless, they all have a richness dip at the equator. And then, so this was very clear that this richness dip at the equator is significant and is not an effect of sampling bias. So then what else is causing this problem? And we tested for a number of variables such as continental shelf and sea surface temperature, chlorophyll. And among all of them, sea surface temperature turned out to be the highest explanatory variable. And this also corresponded, so we can see, so we modeled the species richness against sea surface temperature as a predictor variable. And you can see on the y-axis, it's the number of estimated species richness of all different groups. And on the x-axis, there is this sea surface temperature that goes from 0 to 25 degrees. And you can see that beyond 20 degrees, most of the species richness in all the taxonomic group, either they are just getting stagnant or they are declining. So this was very interesting to find, because so most of them basically, you can say that where we have the highest temperature at the equator, from 25 degrees to 29 degrees in the tropics, that's where the species diversity starts to decline. And this was quite surprising, like why is this happening? Is the temperature already too hot for the species gradient to respond? Because we are talking about these thousands of species that are just not, they're just declining their population in these places, and yeah, so that was a trigger to just further dig into if there is an impact of, this is an impact of climate change, you know. And then I just looked into how sea surface temperature has changed over time in different latitudes, you know, in different regions. And we can see in these graphs, so this is on the y-axis is sea surface anomaly, sea surface temperature anomaly, which is basically the difference between the latest time period sea surface temperature and in the previous time. So how much it has changed. So it represents, this index represents the change in temperature. So we can, and the zero represents there is no change. And we can see that in the Arctic, in the northern temperate, and also in the tropics, the temperature has been, and also like to an extent in the southern temperate as well, up to minus 52.5 degrees. The temperature has been consistently rising since 1980s actually. And this is quite a lot of rise in temperature. The next part of the research was to figure this out, if over time this species diversity gradient has changed. And so then I just modeled the species richness against latitude in 3 different time periods, which is from 1955 to 1974, one time period, and 1975 to 1994, and 1995 to 2015. So the green color represents the oldest time, and the red color represents the latest time. And we can see that in both pelagic and benthic species, we see there is this, the dip was less, but then in the latest time period, the dip has gone much deeper, especially in the pelagic species. Whereas in the benthic species, there is kind of a very significant, there's a shift towards the northern hemisphere. And you can see that especially for the benthic ones, the green, from 1955 to 1974, the species richness has been literally unimodal, but it has changed over time, especially after 1975 here. And this kind of pattern corresponds to this increase in temperature over time, and it was highly correlated. So we can say that the species are actually are moving. Either they are moving to the higher latitudes because they cannot tolerate these warm temperatures, or they are just declining in these tropical latitudes. And there is a mixture of species, you know, so, and it depends highly on their physiology, if they can tolerate these temperatures or not, or if they are very mobile and they are adaptable, and if they have wider thermal tolerance, then they are free to move, and they can also be found in other places. And there are also evidences, we will see later, that species have found moving, or they are predicted to be moving in the higher latitudes. And also species which are very stenothermal, which have like very narrow range around the equator, they are just probably declining, you know. So maybe we are looking at some kind of local mass extinctions, or just this massive movement of species from their native habitats to somewhere else. So, but this was the very first time that we showed on data, especially using the raw data points. And this is not, I mean, this is very closely related to what the data says, you know, it is not a prediction here. So this has already happened. And it's really big, you know. It's also very scary to see that what is happening with the marine ecosystem, and what will be the impact in the future, and how that will affect us, you know. So this just like this study was published in PNAS in 2021, and it sparked a lot of hype around this. I mean, this awareness around this whole marine ecosystem, which people were not really considering. So, and this study was, these graphs were cited by the IPCC in Working Group 2 in their report that was published in 2022, 1 year after this study was published. So this was one aspect of, that we looked into, just the very coarse scale of latitudinal bands, looking at how their species richness, a very simple index, is changing. Then the other part of the research was to see how the species turnover is changing if we go from north to south. And to my surprise, I found that, so this, if you see on this graph, the x-axis is latitude from south to north, and then this dark black line is species turnover. That's the composition of species that changes when you go from one latitudinal band to another. And the dotted lines are basically species richness. So in the species richness, what we found was a dip at the equator, right? But in species turnover, we found that there is a peak at the equator, which means that we find a variety of different species at the tropics. But when we look, so when we go from north to east, south, or even between latitudes, we will find more different variety of species. And it was really mind-boggling for me, because in my mind, I was thinking, well, you go from where you find more number of unique species, that's where you find most number of species, right? In a bigger region as well. And that's what we all expect. But if we look from a bigger perspective of these bands, these regional bands, and we compare, it's not that many number of species. So this was really interesting to see. So in the light of climate change, what is happening, the species turnover, it will increase because species are either moving out, so they are increasing in the high latitudes, but we still find these most unique species at the equator. And this has been because of our geography, because we have a lot of continental shelf and separation between oceans and very unique habitats, which are very, very important and unique to the tropics. So that's why we have a high species turnover here. And species turnover is just an index, because it also shows that when you, it just says that, okay, this area A is different than area B, it could be just because of the loss of species, So turnover can also be high when the species are losing. Or you find completely different species in another, So it could be because of nestedness. It could be because of species loss. It could be because of the habitat loss that is happening nowadays. So we have more patchy habitats, which will hold very unique species, unique to them, So when we go from one habitat to another habitat, we might find different unique species, but doesn't mean that the overall number of species is high. Yeah, so this is what I was talking about. If we look also across longitude, so these are different biogeographic realms that we proposed in 2017. And this is based on species turnover. So as we go across the oceans from north to south or east to west, we find different composition of species altogether, these biogeographic realms. And we can see that they are more in the tropics. This is what I was saying. Yeah. And these, so these are very unique, and they have these biogeographic boundaries, here. And then the other thing was to, like, this came much later, this paper, which was published in Nature, that how the biodiversity has evolved over time, especially from like 500 million years ago until today, along with the continental shelf. So you can see that as the continents have moved and there was more shelf area, and then they kept moving, and then also the diversity, was changing and finding its own patterns and its own stability in the midst of mass extinctions, and they recovered, and that's how we find it. The graph G and H, they are basically we propose a model that based on the previous, these A, B, C, D, E, F graphs or data, how we model it, the present diversity, and how it is already there based on the database that I looked into, OBIS. So it was very well matching with this model. So which means that the models that we propose from A to F seems to be very true along with the continental shelf, how the diversity has changed. And this also kind of explains the different kind of biogeographic patterns that we see here. Yeah. And what is happening, or what will happen with these within these biogeographic regions? So this was a very interesting study that was published recently in 2022 by Brown et al. And they looked into these biogeographic realms, how the distribution of species, how it will change with time, especially under the extreme RCP scenarios where we have indefinite amount of carbon dioxide emission and the global temperature rises above 4 degrees centigrade. And they found out that in these different RCP scenarios, the species from each of these biogeographic boundaries, they are going to move out, which means that we don't know if they can physically move out. We only know that they will find suitable habitats in the other areas, in the other latitudes, or in the other habitats or the other biogeographic range. So you can see these arrows that can that show the direction of the movement of species. So at the moment, we can only predict the using the environmental data, that where the species will find its suitable environmental niche, its ecological niche. But we do not know if the species can actually make it to that point, if they are physiologically so strong or tolerant or are capable of doing that, along with the other associations that they have with other species. For example, their role in the food chain, or competition with other species, and their dispersal capacities. So, yeah. So along with this research, I up to this point until 2019, my work was to just look at these big gradients, in overall global gradients. And then when I moved to Frankfurt in Senckenberg Museum, my work was to find out, okay, let's work with some very specific ecosystem and see what is happening there, So we worked on coral reef ecosystems because they are very important. And we did some, so we only picked a handful of species, 57 species, and we projected them. So we had their occurrences and occurrence data in both cold water and warm water. And these coral reef ecosystem species are basically those species which are fish, mollusks, polychaetes, arthropods, and crustaceans. And they basically were selected because they were living in a very close vicinity. So far, we also have a limited knowledge of the ecological associations between these, between the coral reef species within coral reef ecosystem. So the idea was to just see if they are geographically, living together, or they are more likely to have some kind of association. If not, they still live in the similar habitat or environmental conditions. So will they respond similarly if we project their distribution in the future scenarios of RCP 4.5 and 8.5? So yeah, so for this study, we just collected the global environmental layers from biorecord, which was mainly sea bottom temperature, salinity, current velocity, and in different scenarios from present to future RCP scenarios. And also we used depth as a geographical layer. Yeah, so this modeling is a bit different than the generalized additive modeling that I did in the previous studies. This one was more is a predictive modeling, and this is based on maximum entropy models. So basically what they do, they take the occurrences of the species and they use these environmental variables in the same locations, and then we predict the probability of the species or the occurrence of the species in an environment, which is not geographically associated or close, or it just gives you a environmental niche, not ecological, environmental niche, and that goes into the calibration. Then we calibrated the models and did some bootstrapping to find out the final, the best model. And then the final model actually looked like a global projections, so which was actually showing the probability of species occurrence at a global scale. This is important to do because in ORBIS, if you see, we have point records and we do not have a continuous distribution of species. So this kind of breaks the data. And for this, to compensate that, we were only using bigger grids so that we can say, okay, the species was present in a bigger grid to just cover, to just keep this kind of bias aside or adjust for this kind of bias. But when we want to look into a very finer resolution, then it is important to have a continuous data. So that's why we do this kind of modeling. So we see, okay, this species occurs in this kind of environmental condition. And if this environmental condition is occurring somewhere else or closer to that, what is the possibility of that, probability of that species to occur in that kind of environmental condition? So we get this raster layers, like a map that shows probabilities of the species distribution. And then based on that model, we can also project, okay, present, it looks like this, and then we calibrate it, we validate it, and we make sure that this is not an exaggeration based on expert knowledge. If that species is actually occurring there, what is its dispersal capacity? And then we can predict the distribution in the future environmental conditions, under these RCP 4.5 and 8.5 scenarios, when the temperature is going to be much higher, especially for 8.5, like 4-degree rise. And then the next part of the project was to compare if these maps, the present ones and the future ones, and find out if they are going to extinct, they're gonna show absence in the future scenario, in the same location. So for example, we can see the results. So this is RCP 4.5 and 8.5, and we can see that in 2050, especially let's look at the 8.5 scenario where we consider that the no control over carbon emissions, and by 2050, how the diversity would look like, how many species will collectively lose their native habitats. And we found that especially, so if you look at this 8.5, which is very close to what we are expecting, in the tropics, the species, so there are 30 warm water species, and almost all of them, like 50% of them, are actually going to lose their distribution in the tropics. And some are going to lose their distribution also in the northern hemisphere. And the intensity, the area of loss of the habitats, native habitats, and also the number of species that are going to lose their set of environmental conditions. So these places are going to become inhabitable for these species because they will be too warm. So we are just looking at one aspect of this, which is climate change. We haven't even looked into the anthropogenic threats yet. So that would be even more. And this is just based on the empirical, environmental conditions, if that can exist or not. So if we consider other parameters of species physiology and other things, these might be even more scary results. Yeah, so we can see that a lot of species, especially in the tropics, in the coral triangle, almost all out of, like 29 out of 30 species have shown their loss in their native habitats. Well, these species are also occurring in these parts, in the northern Australia here. So they will still be occurring, they are not gonna get lost technically according to this model, because they will still be occurring here. So when we compared these different models, we found that there was no loss around this part, but there was a very significant loss here. We can compare, look, in 2100, 8.5, it's nearly 50% species here. And then here it's increasing. So, yeah. That was, this kind of like shook me, the result. And I just hope that it doesn't turn true. But the models actually suggest that it is going to get really warm if we have more than 4 degrees of global warming. So, and even in the case of like 4.5 scenario under mitigation, we still be facing losses, but will be much less than what we see here. And this is just over a century, which is not so far away. It's just like our generation and the second generation after us. So maybe our grandkids might be facing this high temperatures. Yeah. And the other thing was to see what if there is any gain in the new habitats, if the species are actually find any new habitats. So unfortunately, especially in the tropical, around this part, they do not find any new habitat. So the species which are already occurring, they will still occur in the southern or in the northern parts. But apart from this, in the Japan coast, a few of them were going there. But basically nothing. And for these northern species which were occurring in the temperate regions, so we saw the loss here, and we can see the green color is basically the places where they're gonna gain their habitability. So which means they will gain their habitable environmental conditions technically according to the models. But if they can move there or not, that is a question. But there are researches which are already saying the arrival, showing the arrival of, noticing the arrival of new species from the Atlantic or the Pacific into the Arctic Ocean, in the North Sea. So yeah, and these models actually show that, and especially for the fish, they're gonna move up into the Arctic because they are more mobile. And yeah, and if we look at the area of loss or gain, especially for warm water species, then we can see the first A graph shows the area of predicted loss for the corals and others, all the species. And then this dark orange color is the RCP, the worst case scenario in 2100. And you can see that basically in all the scenarios, the loss is a lot, and it's the worst in this 2100. And the B part shows gain. So, which I was saying, if they gain a new habitability in the other regions, which is much less if we compare these x-axis, y-axis, in between these two graphs. So this gain is much, much less. Yeah. Only fish may survive to a certain extent, by moving apart or, yeah. And then if we look at the cold water species, so we see that the loss and the gain is basically kind of comparable. And this is very much shown in these graphs, There is a loss in the lower latitudes, but gained in the higher latitudes, especially in the Northern Sea, in the Arctic. So we can expect a high turnover in the Arctic Ocean. But how this species turnover is going to affect the ecosystems, in the Arctic and also in the lower latitudes, we don't know. It's beyond, because we need to know how the ecosystems work, and we still have a very limited knowledge of how the species are working together in association with each other to maintain the ecosystems. And if species are starting to move out, then what impact will that have on the ecosystem? So it might lead to dysfunctioning of the ecosystem. The ecosystems may survive. We don't know that. But from our limited knowledge and research, what we can see is that all the coral species on the left-hand side, from the warm waters, all these representative species of the ecosystem, they are showing unanimously more loss than they gain. Whereas in the on the right hand side for the northern species, some of them are losing and some of them are gaining. When I research about the names of the species, where these species occur, what kind of thermal tolerance or the range they have. So we see that the species that are occurring in these higher latitudes, they have more thermal tolerance, and they can travel longer distance, and that's why they are gonna invade the northern waters. But the species which have very narrow thermal range, which are very specific or unique to their environment, for example, the Arctic species or the tropical species, they might not be able to adapt to this new situation, and those are the ones who are losing. So these are the stenothermal species with very narrow and very specialized thermal niche. And even in these in the right hand side, you can see the ones which are losing more, they are the ones who are occurring mostly in the Arctic, in the north. So yeah, so this was the species distribution modeling that tells us the insights of the future. But we also have a lot of limitations in these empirical models because they do not include physiological information. We don't know if species are physiologically, how they're going to adapt and how they work in association with each other. So my current research work deals with that, and especially looking into the life stages thermal tolerance of these data. So for example, it's a very complex figure, but let's break it down. So we look on the right-hand side with the graphs that we are familiar with, these bimodal, unimodal graphs across latitude. So I started from there, then went into a bit deeper to look into much finer models, found there is a dip because the species are just not, making it up to new habitats, and then we assess their habitat suitability, and then comes the different life stages. So it is very interesting to know that species have different life stages, and all these life stages have different thermal niche. So some of the life stages, for example, in fish, the larvae and adults were found more tolerant than the embryos and spawners. So which means that if the temperature rises, then these spawners and embryos are the ones who are going to face the most vulnerability, and they probably will die first. And my work at Alfred Wegener Institute is just looking into these bottlenecks and finding out if the dip at the equator that we found is because of these vulnerable life stages, as what Fleming et al. Has found here. So, yeah. And then, so in this graph, you can see that as we go from south to north, in the graph B, and the adults and spawners. So the blue color is sea surface temperature. So sea surface temperature is very close to the maximum thermal tolerance of the spawning fishes, which is exactly the place where we found this dip at the equator. So it is highly likely that these vulnerable stages are the first ones so these are the very productive marine zones, for marine life, and we might lose them first. So the short summary is that we still have a long way to go, but what knowledge we already have, we can use that to implement certain protected areas, or do more strategic resource management. And this is the importance of biodiversity climate change research, because then the stakeholders can use these high resolution models to identify the regions of high risk, and also work on how to protect them, or what are the other factors which are affecting them. And also not just that, but also keep a very close eye on how these ecosystems or the species diversity has been changing. And then we can see, okay, if this is good for us, or if we need to work something more, we need more data basically. But we use these models to already conserve our biodiversity. And one way of doing it is to implement marine protected areas. These are distinct zones for which protection objectives are set, and they constitute a globally connected system for safeguarding biodiversity and maintaining marine ecosystem health. And basically helps in conserving the biodiversity, habitat protection, also fisheries management, and scientific research, and recreation and cultural activities. So we need a very good ways to find out how to design the best, way or the most effective marine protected areas that not only conserve our biodiversity, but also be very efficient for fisheries management, so the local communities do not suffer from that. I will show 2 examples for these protected area, like how we propose the protected areas using biodiversity information. So this was the study published by Irawan Assad et al. In 2018. And so we can see the coral triangle and the distribution of threats, anthropogenic and thermal. So the red color shows higher threat, and the blue color shows lower threat. So we can also already see that places where we have higher anthropogenic threat, we don't have so much of stress from thermal, and nevertheless, the temperature is increasing and it will be a threat. And then he proposed use these biodiversity information, to propose the MPAs up to 10%, 20%, and 30%, so in this graph, we can see the black dots are actually the already existing MPAs. And then the red ones are the ones that conserve like 10% of the area. And they are suggested to be very efficient, using the biodiversity, in information plus the threats. So this was proposed in 2018. And then there is this new research that is recently published in 2023 by Jefferson et al. And they worked on the data from New Zealand, and they looked into the distribution of marine protected areas around New Zealand. So this is the current protection in the left graph, A. And then they just looked into, okay, if we just look into the total fishing catch, and if we just look into the maximum, like the ones which only the fishing catch of the fish which are of high quality. So, and then also consider the biodiversity index. So they used a species distribution modeling, like a prediction modeling, to have this continuous data, and they got more continuous graphs here. And we see that in scenario 1, where they just were looking at the total fishing catch, they proposed these marine protected areas in blue color, and the red color here shows the overlap with the already existing marine protected areas. So we can see that there are a lot of areas that don't need protection, but they are still under protection. And then there are areas that need protection, but they are not under protection. So, and then they also find out that that 30% of protection of EEZ could cover more than 86% of threatened species and biodiversity, and also maintain 86% of fishing catch by weight. I'm sorry. So, so we can see here that and they actually also not just consider the biodiversity, they also consider the threatened species. They also considered the most protective zones, So if we collect more information about how these ecosystems work, we may find that we might save some areas there where we can still fish, but maybe we have to protect it for certain time, so that it kind of regenerates. And then so we also have to be flexible to move these things. And also knowing having this information that how the species are moving. So we also have to be think about, like, if the current MPAs are actually working or not, and also their assessment over time, to see if they are still efficient. So yeah, so we need more these models, they prove very efficient, but also we need the information knowledge from a local community to make the decision, along with the stakeholders for the resource management. And we need more monitoring data, basically, so we can just observe and see how these ecosystems are changing and make most out of it, and be in harmony with nature and not against that. And I think that will just save us and our humanity. And also I also always feel like it's not just like we are at the stake, not the animals. I mean, the nature is always, evolving, and there were also mass extinctions before, but it has just bounces back. But it's us who I think are at the stake, and we have to take care of ourselves. And to do that, we have to first take care of our surroundings and our nature, and be more mindful of how we are utilizing our resources. And I would like to acknowledge all the research institutes that have given me space to work, and also my collaborators and my supervisors. And here I end my lecture, and thank you. I hope I didn't take too long, and we still have time to discuss about this. Thank you. And thank you so much for inviting me and giving me this space to talk. Thank you.
Okay. Maybe the participant has any question for Dr. Chaya Chaudhary. Thank you, Dr. Chaya Chaudhary, for your presentation. To all the participants, okay, Mas Malik. Okay, Mas Malik.
Okay, thank you, Dr. Chaya. This is very interesting research, but about the climate change, we can imagine that if climate change is occur and coral reef become extincting, then of course the coral reef in Indonesia will be just a memory, yeah, because Indonesia have rich and biggest coral reef in the world. And you come to Indonesia first, doctor?
I
No?
really want to visit Indonesia, really, but I haven't got this luck until now to visit Indonesia.
Okay. Hope you can come in Unsur and you can look at Indonesia research. And during the presentation, I also read this journal. I will just can me to be the host.
Oh
So
yeah,
I
sure.
can,
I'm sorry.
I
Yeah.
can explain.
We'll just stop sharing. Yeah.
Okay, wait. I just want to tell you for our student that during this presentation, I already read the Dr. Chaudhary journal. Okay, this is a journal that represent to doctor. Okay, if some of you have the question, any question, you can raise hand, you can unmute your mic, or you can send the text in the box of chat room. Okay, and wait, stop there.
I think we have someone raising their hand. Pak Malik.
Okay, just open the video and you can share what you ask. Okay, what's your name?
Okay, my name is Mufida Romatunisa, and
Okay, Mufida. Thank you.
okay, my question is for Dr. Chaya. What is the biggest problem or obstacle that you face during the experiment, and how you handle it? That's all. Thank you.
Okay, first question. And I just want to say that we would like to also share voucher to the most active participant who give the answer, give the question, and the other lecture we will contact you after the event. Okay, the second question. Okay, before the second question, question from Mufida. Dr. Chaya, you can get the question?
Yeah, if correct me if I'm
Okay.
wrong. So the question was, what is the biggest challenge that we find in the experiments and modeling? I can answer for both. So for experiments, the biggest challenge is that we cannot replicate the. For example, if you want to see how species are, adapting physiologically when we change the temperatures in our experiment. So one big challenge that I'm finding is what we do, we expose these species to a very high temperature at a very short time, which is not happening in the real world. So I'm analyzing a lot of experimental data, and I see that they just raise 2 degrees in 6 hours. And then next 6 hours, another 2 degrees to see to what temperature the species is physiologically going to tolerate. Of course, it has more energy, and maybe for the first time it might do that, but it doesn't because this temperature is increasing for sure, but it's not increasing so fast, So this is a challenge because it doesn't replicate. This is a challenge because we do not have so much fundings per project to do these long-term experiments where we raise the temperature slowly, because in the real world it is increasing, and even a difference of 1 degree, 0.1 degree makes a big difference if we look at like 30 years time scale or something like that. In modeling, I would say that there are many times where we have to decide the compromise on the resolution of the data because we do not have enough data. And the more data, the more observations we will have, we can do more precise modeling. So I hope I answered the question.
Okay. Mufida, you enough the answer from the Dr. Chaya for your question?
Yes, that's enough. Thank
Okay.
you.
Okay. I'm just right. The 3 point in this discussion. Firstly, is understanding the species in the diversity is very important because their shifting may predict their pattern in the future. And the second is the tropical has higher diversity, where areas the higher latitude reduce the species diversity. And the third is the higher the temperature was appeared at the equator line, the pattern of the species is correlated with high temperature condition. The species might be moved to the other region or cannot cope with the internal condition. Maybe that's it, Dr. Chaya. Okay. Any student will ask the question? Okay. We have any time to discuss?
Maybe I will give a question for Dr. Chaya.
Okay. Thank you, Miss Ruri.
First of all, it's a very interesting talk, Chaya. Thank you for joining our event today. And I would like also to ask you related to, you mentioned that in your study there is an obstacle that you haven't really included the physiological aspect in your model. So, what will you do in the future related to that, and how you're going to match the physiological aspect with the modeling aspect that you've done? And also you mentioned that currently you're doing the modeling also with the larvae stage or the stage of the species. Is that really also correlated with the physiological aspect that you would like to see? Yeah, thank you.
Thank you so much, Ruri, for the question and also for the compliment. Yes, that's the thing. So until my modeling in coral reef ecosystems, it was all empirical and it did not include physiology, except the dispersal capacity of the species. And this is a challenge, how we incorporate these experimental values or these curves, where species is very happy in this optimum temperature and then it declines, because this thermal threshold, could be very high in the experiments and maybe not so, you don't see it in the realized niche. So it's a big challenge, and I'm still trying to figure out how to use this information. But one way of doing this is what we found is to just find like a pages temperature where we have the realized maximum thermal temperature, realized temperature range, and also then subtracted from the critical thermal range where the species starts to decline. So it's like something between the maximum thermal range in the realized niche where it is found, and then before the critical niche in the experiments where it declines because of hypoxia. So maybe work with those temperatures. And including the reliable information. So what we were thinking is to, there are two ways of doing that. And I was just in a meeting yesterday discussing this. So one way is to just model especially differently for larvae, adults, spawners, have different models and then compare if they have any overlap between, and then we can use the experimental values just as like a constraint in the model. So we cannot really put the index because it's not an index, no, it's just one value. So we can use it as a constraint to see to not that the model doesn't over exaggerate, or we can just do the modeling for the species and then compare the results, overall, and then later on compare the results. Yeah, something like that. So we're still figuring that out, and hopefully we find the answer. There is a very limited literature. People either use mechanistic models or they use empirical models, and we are trying to make the most of both of these things. So it's a challenge still.
Okay, thank you, Mrs. Ruri, for your question. And for our student, there is any question? Because we have voucher for. Okay. This is the question. Okay, I will read some question from Heri Irawan. Can this MPS be implemented in urban areas, considering that MPS can be natural fortresses that protect the coast from tidal waves, storm, and floods? Okay, Dr. Chaya, maybe you can see and read the message in the chat box. Okay, you got it?
Yeah, you can put MP anywhere except that it's basically in the marine, coastal region, which is yeah, why not? And but MPS is not just protecting from the coast from tidal waves or storms. It's basically protecting the biodiversity. It's much more to that, protecting all the natural resources and make sure that there is no commercial fishing, or sometimes there are different levels of MPAs. So based on the assessment of how the situation is and how much we need to protect that area, we can apply these different levels, So there is a level where nothing can be done, no fishing, no recreation. But there are also levels of MPAs that can be where you can do recreational fishing and also some bycatch fishing, So it depends on the situation. But I think that as long as it is in the marine realm and not the terrestrial, you can suggest MPA, yeah, based on the data and monitoring data, basically.
Okay, thank you, Dr. Chaya. How, Mr. Irawan, you can get the answer from the Dr. Chaya? Okay, it would be very good, if you, doctor, you can come to Indonesia and you can research here about MPAs, because Indonesia have a lot of MPAs, because Indonesia have coral strangers area. We have Raja Ampat, we have Bunaken, the home for millions of coral reef in the world. Yeah. Okay. Some of you, the student, you can ask the question. Okay. Hesti Puspitasari already raised hand. You can open your mic and open your video. Time is yours.
Okay. First, thank you for sharing your knowledge to us, Dr. Chaya. And then based on your explanation, I have some questions. First, why the most effect of climate change is happen in Khatulistiwa areas? That's it.
Okay.
Sorry,
you
can you repeat it? I couldn't understand. I think the voice is echoing. Sorry, can you repeat it, please?
My question is why the most effect of climate change is happen in Khatulistiwa areas.
In
About
the
climate change in Khatulistiwa. Khatulistiwa is equator, right?
Yeah, tropical area.
uh, because it's just hot and then it's getting more and more hot, you know. So it's I mean, because of these carbon dioxide emissions, you know, so then it's global warming, and the effect is highest at the tropics that you see, but also it is also higher in the poles, you know. So, and also it's because of the circulation of the air, how it goes, the wind, and then how the oceans work. So then it brings more warm, you know, air, and tropics have always been the most stable warm temperatures, so they don't vary so much, and the seasons, you know, the temperature don't vary over the year if you see. But in the higher latitudes, you see there is much more variation, so there's cooling down and then going up. But then these areas are also getting warm days, you know. In Germany nowadays we are seeing the temperature is getting up to 40 degrees. Where I grew up in India, it used to be like 28, 30 degrees maximum when I was much younger, but now it shoots up to 45 degrees. And the effect is much more worse in the tropics, actually.
Yeah. Okay, thank you, Dr. Chaya. And I think the terms of climate change, global warming, is no longer. And now the scientists and the people just using the boiling up, yeah, Dr. Chaya, boiling up to describe the climate condition.
Mm-hmm.
Yeah. Okay. Maybe it is the last question from Nani WRC in the box. Okay. My name Nani. Thanks for Dr. Chaya. Could you provide example of successful marine protected area initiative around the world? Thanks.
Well, there was this in 2020, there was this initiative by COP, I think, to protect like 10% of the EEZ. And I think in Europe they are doing that. And now it is the goal is to protect 30% by 2030, which is a very you know, is a very ambitious goal, but they are trying their best. And yeah, this is all I'm aware of. I'm sorry, I'm not directly working into the MPA area, so I have a limited information on that, but I'm mostly onto in the modeling side where we provide the data. So
okay, some of you will give the question again. Okay, Saidah Nurul. Okay, time is yours.
Okay, thank you, Mr. Malik, for the time. And thank you so much for Dr. Chahya. Thank you for sharing this very interesting experiment. My name is Nurul. I have a question. How can data and modeling technology be optimized to increase accuracy in predicting shifts in species distribution due to climate change? That's it.
yeah, there are many different ways of optimizing. So for example, we have used parameters, and we calibrated the models based on the parameters. And that is basically, you look at the R square of the model, especially in GAMS, and see the significance value of that. And then for the max and prediction models, we use ROC curves, and these curves you can see how closely are they related to the raw data points, you know, or if the prediction is going in that direction or if it is going in another direction. So you kind of know like if you can trust this model based on the distribution of the points that you get afterwards. And then there are also other ways, especially for this Maxent modeling. I use this R package, KUENM. It's a really good one. So you can look into for that, and it gives you this very nice cross validation. So what it does, so you produce the model, and then, for example, you get these environmental niche somewhere in the very north, where probably the species is physiologically not possible to go, right? So then you know that this is an extrapolation, okay? But then there is also like the probability could be from 0 to 100%. So how much can you trust? So then you define a threshold based on your discretion. Let's say if I find a place which is 3% similar to the original environment of the species, right? So you know, okay, you can trust it. But anything above 3%, you say it's an extrapolation. So then you have to make a like a discrete decision based on your expertise, whether you go for 5% or 3 or 2. It's a very theoretical in the end, you know, but you have to decide based on your expertise. But then you can tell the model and you check for that 5%, how does it look like? 3%, how does it look like? And that kind of helps you to narrow down these extrapolations. Yeah.
Okay. Thank you, Dr. Chaya, Saida, yeah. And then maybe the last question in the box. My name Putra Juvaldi. I want to ask about in Indonesia now the weather changed so quickly, especially in Purwokerto. Example, at the 1 p.m. it's very hot, and at 3 p.m. it is raining. How do biota organism adapt to the fast changing temperature to survive in their habitat? Thank you, Dr. Chaya. Okay.
That's a very interesting question. And I'm also wondering how, I think sometimes plants go confused when it's here also, like when it's warm, all of a sudden they start blooming. And then next day it's just the temperature drops and they drop all their flowers. So I don't know. I can't answer this question. But I think if they you see these mass mortality, especially during these days of heat waves, when a lot of like fish, they just die on the shore, or the birds, you know, they are they, because of the change in direction of the wind, they just go in the wrong direction and they get confused. So they are suffering. They are really suffering, and only the ones who are very tolerant species, they will survive. So I think if this continues, and if we are going to lose our uniqueness, there will always be some species in the ocean or on the land. But especially for the oceans, I can say what I see from the models, that we're going to lose the uniqueness. We're going to lose the, how do you say, the colors, you know. We will have more general species which we find everywhere. So yeah.
Okay.
But that's
Thank
a
you.
very good question. Thank you
Yeah,
for asking.
good question from Putra Juvaldi. Okay, in this room, I will greeting to our lecturer, Mr. Dr. Maria. She stay in Korea for now for study. If you have some question or something, you can ask to you can share to us about the modeling of climate change and MPAs. Mrs. Maria, you can hear me?
Yes, thank you. But I think Dr. Chaya already explained everything clearly, and you did really a great job, Dr. Chaya.
Thank you so much.
For sharing the information about how to modeling the biodiversity, and it's very, very interesting. And in this lecture, I think some students also, they are taking the lecture of the conservation. So it's really, really, you know, give a new insight for them. So I just want to say thank you to Dr. Chaya for sharing your knowledge to our student. Thank you.
Okay, thank you, Dr.
so
Maria. Okay, ladies and gentlemen, finally we come to the end of the presentation. We would like to say thank you very much to the presenter, Dr. Chaya Choudhary, for the interesting presentation, and also to all of the audience for your active participation. Hopefully the presentation will be beneficial for you, for everyone. Thank you for your attention. Okay. For Dr. Chaya, we will like to give the present certificate for you. And okay, this is certificate for you. Okay, we can applause for Dr. Chaya Chaudhary.
Thank you so much. Thank you. It has been a great experience, and I'm so honored and so humbled to speak. And thank you so much for giving me this platform. I'm so humbled. Thank you so much. Thank you so much, Ruri, for reaching out. And yeah, I mean, this information is so important. I mean, I also feel sometimes, you know, it's heavy information, but I also feel it needs to go out, and we need to be aware. It's just we have to adapt to this change, and just be kind, you know, and just the species are also adapting. They are also suffering, but they are also adapting. So we can also do something similar and be kind, you know. That's it. Thank you so much for giving me this platform.
Okay, thank you again, Dr. Chaya. It was pleasure to have you with us. So this conclude the webinar. Thank you for attending. We hope you have learned and enjoyed this presentation. Okay, Miss Dika, it's yours.
Okay. Thank you, Mr. Malik, for lead our main session. It's just a great time to have a speaker as well. A special thank you, Dr. Chaya Chaudhary, the valuable and wonderful lecture for all of us. And next agenda, we would like to present certificate to our moderator, Mr. Abdul Malik Firdaus, Master of Environmental Science.
Yeah,
This is
thank you.
this is the okay. Okay, thank you to Mr. Abdul Malik. And then, ladies and gentlemen, I would like to remind you to fill attendance form and check the certificate. Okay. And then for active participant, we'll give a voucher after this event. Ladies and gentlemen, this is the last agenda. Ladies and gentlemen, this is the last agenda. As the master of ceremony, I would like also apologize if there are many mistakes during this public lecture. I wish you have a pleasant lecture. I suppose that would be all, and thank you very much. Good afternoon, and assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mr. Chaya, Mrs. Chaya.
Yeah. Bye, contact you soon. Bye. Have a nice day.
Ya.
Terima kasih Bapak Ibu semua.
Terima kasih untuk semua mahasiswa yang sudah join.
Ya, udah mulailah.
Hadiahnya ditunggu diberikan oleh panitia ya.
Ya, nanti dikasih terima kasih nanti hadiahnya.
Terima kasih ya.
WA.
Tadi dicatat ya,
Ya,
yang
tadi
sudah
udah
dapat voucher.
Sudah dicatat.
Voucher token listrik ya apa?
Iya. Iya, udah dicatat tadi. Oke, nanti tinggal dikirim langsung nanti setelah ini. Sama
Izin
panitia.
ya, Pak Dika semuanya.
Oke, iya Mas Malik. Makasih ya Mas Malik. Assalamualaikum. Iya,
tadi yang dapat berapa? 5 orang ya Mas Malik?
Iya, kayaknya 5 orang.
Oh.
Ruri sama Mas Dery nggak usah. Berarti 5. Mufido.
Kalau Bu Ruri vouchernya harus paling spesial loh.
Nah, ini yang berarti yang nggak usah dikasih Bu Ruri sama Mas Dery. Nurul, saya
yang lainnya
Asaidah, Nurul sama Mustafa. Sama kok Mustafa?
Masuk. Oke, see you ya. Terima kasih semuanya.
Terima kasih semuanya.
See you, Bu Nani. Mbak Nani ya. Oke, see you.